obviously going to be a problem, because if the blood is not in the circulatory system, then it's not going where? To the cells, and if that's not happening, the cells are gonna die, and that's called necrosis is what happens in a lot of the cases, right? So without the venomous snakes that we have, they have a lot of these things, right? So if you get bitten by a copperhead or a cottonmouth, what's gonna happen is these digestive enzymes are gonna start breaking apart proteins, including the structural proteins that hold together vessel walls, and the blood is just gonna leak into the tissue. The cells are gonna die, and then ultimately that's, you know, for a person, it's not deadly, but it could be destructive, right? So a lot of times if you get bitten on the tip of the finger, sometimes the tip of the finger has to be removed because the cells die and you can't recover from that. So the venomous snakes that we have in Tennessee, at least, are mostly that. These kinds of proteins that are going to interfere with blood flow, and they break down tissues. Some of them are enzymes that affect the nervous system. And so acetylcholinease is an enzyme that will ultimately interrupt nerve impulses, and obviously that's going to be important because that's important in respiration, heartbeat, and all that kind of stuff. So our, native snakes in Tennessee don't really have a lot of that, because if you have a lot of that in venom, then that's really going to have some big influences on the nervous system, which can shut down respiration and circulatory system and all that kind of stuff, which will ultimately lead to death pretty quickly. But most venoms have some of those, and again, it depends on what kind of animal we're talking about, but they all have those things. And then some of the elements of venom are just polypeptides, so chains of amino acids that do damage. And those things, again, are going to be most specific to the nervous system. And so, of course, different types of organisms have different structural polypeptides that do different things. But mostly they're going to be interfering with the nervous system, which is always going to affect the respiratory and circulatory system, and that's essentially what causes death in the prey item. And many times they work pretty quickly. And another thing to think about is most of the time these animals are attacking relatively small prey items, right? So not humans. And so it's going to have a rapid effect. A lot of these animals, like rodents, have real high metabolism. The higher metabolism is, the faster that's going to take effect, and the quicker the animal's going to die. Cobra toxin, of course, is pretty typical of those animals in the crates, cobras and things like that. So the polypeptides and their venom affect cardiac muscle. So that's going to be a really important one, right? So if you shut down the heart, the pumping mechanism, then all the cells are going to die pretty quickly because they can't get the things that they need. Viperitoxin is more typical of the pit vipers, and for us that would be Crotality, like the rattlesnakes and copperheads. That has a variety of effects. There's different types of polypeptides that do different things. Some of it's going to affect the central nervous system, the brain. Some other forms are going to affect the cardiac muscle or nerve impulses going to from the heart. In addition to polypeptides, there's some other elements that are typically found in venom, including ions and other elements, I guess, that act with the other things. And so you guys, when you were in freshman biology, you did some stuff with enzymes and how they work, and cofactors was one of those things. So you've got these enzymes that have functions, and you can do different things in the environment to affect them. One of the things you can do is take away some of the other elements that are necessary for the enzymes to work. And so a lot of these are components that aren't either polypeptides or particular types of enzymes, but they are necessary for the other things to work. And so without those, it won't work. And so they're involved in a lot of things. Some of them are going to inhibit the immune response. And so obviously that's going to be important because immediately when there's a foreign body in our circulatory system, our immune system recognizes that and then ultimately tries to destroy it. So if you inhibit the recognition of that, then it is allowed to move around freely in the organism and do all kind of destruction. So that's obviously important. So that's all I wanted to say about the typical way that these organisms eat. So if you think about all the amphibians and reptiles, most of them are just grabbing prey items, crushing it, and swallowing. And then you've got some that are constrictors that are a little bit more specialized, and then you've got some that have some type of venom delivery system, and that applies to the vast majority of amphibians and reptiles. Yeah.
So what's the reason for having so many different types of venom in general? Does it have to do with, how is there some that digest tissue proteins and some that attack the brain? Like where do you get the
Well,
differentiation?
what I was summarizing basically,
Right.
like all the different types of, or mostly reptiles that have some type of venom that they deliver. All of them have a variety of components to it. They're not only one molecule, but mostly what you see is they don't all, like each species doesn't have a whole variety of different types of venoms available to them. They have one type, and it's essentially, if you go from individual, it's not variable within a species. And I'm not sure if I'm answering the question. So the variation comes when you go across species. But the advantage to having multiple of those things, because some of them are gonna act to enhance the enzyme. And so, if you've got this molecule that helps the enzyme work, then it's gonna break down blood vessels quicker, and that's gonna cause the animal to die faster. So it's all adaptation to making the prey item die faster. And so it's open to natural selection just like everything else. And so, if you have only one specific thing that is your venom, then your chances of being successful in a different prey item would be less. And so, adaptation is gonna help that, yeah.
So obviously it's not venom, but not just snakes either, but other animals have like components in their saliva that kind of do some of the same things that some of the venoms do, like bend the blood and things like that.
Sure. Yeah, I can think of off the top of my head, bats, like vampire bats secrete stuff, leeches secrete stuff that, that disguise the fact that they're, letting blood come out of the
Ticks too, I think.
What's that?
Ticks, don't they send?
Ticks,
something?
yeah, so there's a variety of organisms that have some type of secretion that's gonna have an influence. Obviously, that's important, natural, the animals that can cause it to happen quicker or cause it to go unnoticed are gonna survive better than the ones that, oh, crap, you're gonna pull it off or scratch it off or whatever the case may be. But yeah, animals do a lot of different things, and amphibians and reptiles are no different. So, yeah, any other questions? Okay, and then also there's other animals that just, all animals have bacteria in their mouth that can do some serious damage if it gets into the tissue, but not in terms of, it's not really a defense mechanism, because it's not really, the biting is the defense part of it. It's not really gonna have an influence on what happens later, if it's just infection, so that's pretty typical too. Okay, so the next bit I wanna talk about are some of the specialized types of feeding, so not very many organisms have this, so I would include that in what I would call specialized feeding mechanisms. And so some of the aquatic organisms, or most of the aquatic forms of amphibians, I would say mostly amphibians, and I guess some reptiles do, have suction or inertial suction feeding. And so essentially what's happening here is the animal, whatever it might be, just kind of expands the buccal cavity, creating low pressure, and then opens the mouth, and then because it's low pressure and there's high pressure on the outside, stuff goes into the mouth, and that's called inertial suction feeding. And so most, that's how fish feed, that's how aquatic stages of salamanders feed, that's how adult aquatic animals feed. That works great if you're in water, right? Because water is dense, it's not squishable or movable, it doesn't move very easily, but if you can get it to move, it carries everything with it. And so that's typically how aquatic animals are going to feed. And so ultimately you just have a kind of a rapid reaction. I'm not sure if I can get this. Let's see what happens if I put it on. That used to be a movie. Let's see. Okay, just Google inertial suction feeding and you'll see it. The basic idea is that it's common in aquatic animals. Okay, so that's not really specialized. So I didn't really describe that very well. So it's not specialized. How come? Why is it not specialized? Why is that a bad choice of words?
So all the aquatic creatures basically use it?
Well, it could be all the aquatic is specialized, but
Because it doesn't require any kind of modification.
from Ancestral, yeah, so it's the ancestral form, so it's, you know, it's not, doesn't really go very well with the term specialized, just because they inherited it from their common ancestor. But specialized in the sense that it's different than most, I guess, is what ultimately I was trying to say. So, another type of specialized feeding in amphibians and reptiles is some type of protrusible tongue. It's fairly widespread, and so it's a kind of an accessory to ingestion. So most of the animals that have protrusible tongues also will grab if they, you know, if they can. They don't have to do that, but they often do. So it's pretty widespread among amphibians, and there's some reptiles that have protrusible tongue. So you guys, I guess, can probably imagine what I'm talking about. So the general idea is that the tongue is protruded from the margin of the mouth, and it comes in contact with the prey item. Typically, the tip of the tongue has some kind of secretion or physical morphological characteristic that makes it sticky or adhesive, and so that's going to ultimately bring the animal back into the mouth cavity as the tongue is retracted or rebounds back into the mouth, depending on the kind of animal. So that's basically what's happened when we talk about protrusible tongues. But there's different types of protrusible tongues. They're different in amphibian, or like if we look at amphibians, it's different in frogs than it is in salamanders, and so a little bit about that. And so if we think about what's happening in frogs, morphologically it's different because their tongues are attached to the front of the mouth for the most part. And then 95% of the tongue is posterior to that. And so what they're doing is they're flipping the tongue upside down, essentially, out of the mouth. They're flipping the back of the tongue. So here's just a bullfrog that's got the mouth open, and so here's the tongue, and the tongue is attached right here near the front underneath. It's not at the very tip, but it's almost at the tip of the tongue, where the tongue is attached to the floor of the mouth. And there's a cartilage there that underlies the tongue. And then the back of it is free. And you can see in a lot of these, they have some type of tabby looking structure on the back of the tongue that helps kind of surround the prey item and bring it back. Yeah.
So is that because, so they flip it out backwards, so when you pull it back in, it throws it to the back of the throat?
It doesn't, I mean, it's mostly getting it into the mouth is really, it's all about that. And so just because of their morphology, what they'll ultimately do is flip the tongue out, not like the top of the tongue is gonna hit the animal, and then when it comes back, then they're just gonna crush it and swallow it. So it doesn't really, put it in the esophagus, it just puts it in the mouth cavity. So for frogs, as I said, there's like a cartilaginous rod that's underneath the tongue running this way. There's muscles attached to the underside of that. All certain types of hyoid muscles that are going to be involved in tongue protrusion. So in this case, the different set of muscles just kind of pulls the mouth down, and the tongue is kind of stiffened. The muscles within the tongue stiffen the tongue, and then the jaw is pulled downward, and that's going to kind of start the process of momentum gathering, and that's ultimately going to result in the tongue being kind of flipped out. So it's not just down, but it pulls it forward a little bit and then down, and so the tongue starts to move forward and then down real quick, and the back end of it flips out, essentially is what's happening there. So the momentum in this case pulls the tongue out. The tongue is relatively elastic, and so that force is going to stretch the tongue out. And most frogs don't have protrusible tongues that are at great distances, right? So they're not going to flip like a chameleon. It's nothing like that, but it does help them get the prey item in the mouth cavity. So ultimately, this is kind of what happens. So the tongue is pulled forward and down really quickly. The back end of it flips out and comes in contact with the animal. So here's just another kind of photograph showing what happens during that process. I don't know what happened. One of my videos didn't load.
You gotta enable the media to load.
Oh, okay.
You gotta go back and click on it on the top somewhere.
You guys do that when you're watching the frog feed. It's pretty cool. And of course, it's all super slow motion, so you can really see the tongue move forward a little bit and then pull down, and it comes out. So if you guys check out some videos, it's pretty neat. And then of course the tongue is pulled back by another set. They have a whole series of muscles in the buccal cavity, hyoid muscles, various forms of the hyoid muscles, and that's gonna be involved in bringing the tongue back in, ultimately into the mouth cavity, and then they just crush it and swallow. In most cases, in nearly all frogs, the orientation of the tongue flip is along the long axis of the body, so they can't really flip it sideways, and so they have to be in front of. And two things are happening. Most of these animals, their prey detection is best when they are directly in line with the organism. Their vision works best, their depth perception is best when they're turned. So most of the time they're doing that anyway, but in combination with that, their tongue only works directly in front of them in most cases. There are some that can go side to side. So there are some species of microhylids, which is what? What's that? Narrow mouth toad, yeah, narrow mouth frogs or narrow mouths. So in some, not all species can do that, but in some, they can make an arc, basically. So you can see that they can really extend the direction from which they're flipping their tongue, from all the way from this side, all the way to the other side. So almost behind them can they catch prey. That's unusual, right? Most of the time, they're gonna be oriented that way, but they can make an arc all the way around. And so if you watch a frog feed, typically they're gonna turn their body so that they're directly in front of the prey item. So Google that one too. Okay, so in salamanders it's a little bit different. So the tongue morphology is different. The tongues aren't attached in the front, so that's different. And there's some variability. So the typical salamanders, like the ones that we see around here for the most part, the ones that, like Epistomatids, they have protrusible tongues, but they're not extensive, essentially. And so this is, I don't know what this one, this might be like a stream-sided salamander or something, but they have fleshy tongues that they can stick out, but it doesn't come out very far. Ambystoma have protrusible tongues to some extent. It's still kind of sticky, so it helps them get prey items in the buccal cavity. But in others, it's extremely projectile. So this is a Plepidontic salamander. You can see, how far that extends out of the mouth cavity. The animals that have protrusible tongue also happen to be lungless, and the reason why that's true is because the hyoid apparatus, which consists of bony elements and musculature in the ancestral species forms, is used for buccal pump respiration. Right? So combination of those bony structures or cartilaginous structures and muscles are used to drop the floor of the mouth, push the floor of the mouth up, and that allows them to push air in the lungs. But you can't do that and also have extremely protrusible tongues at the same time. So the same structures that are involved in those things are ancestrally involved in respiration. And so there's another example popped into my head. Anyway, so the animals that can do that are also lungless, because you can't have buccal pump respiration and also extensively protrusible tongues at the same time. Oh, it's like running, when we're talking about the apneustic periods in lizards. So the muscles that are used for respiration are also used for running, so you can't run and breathe at the same time if you're a lizard. You can't be a lunglet, or you can't be a lunged salamander and also have protrusible tongue. So there's fairly limited number of animals that can do that. Here's another really cool video that shows this Plepidontic salamander eating a termite. But you can't see it unless you Google it. So unless you guys Google that. Y'all know about Google, right? Okay. Reptiles, again, same basic idea. The mechanisms are a little bit different. The hyoid, which is, again, it's a combination of bony elements or cartilaginous elements and muscle tissue, essentially pushes the tongue outward. And then the momentum from that is going to stretch the tongue out. And so ultimately it can be quite long. And so a lot of animals have tongues that can be pushed out of the buccal cavity. Geckos, of course, use them to lick their eyeballs, also to help catch the prey. I used to have a gecko. We would watch him lick his eyeball all the time. That's kind of fun. And then some of them are going to be, protrusible, but not to a great extent, which includes things like geckos and iguanas. And then you've got the chameleons, which have extremely protrusible tongues, as much as twice the body length, basically. Their tongue is twice the body length, and so they use that obviously to catch prey. And you know, we talked about their vision too, so they have eyeballs that can kind of rotate, and they can, and then, you know, turn their eyeballs so they're all facing. They have good depth perception. Of course, you have to have that, right, to be successful with that particular mode of prey capture. So it can be pretty extensive. Here's another awesome video showing somebody feeding a chameleon, but you guys can't watch unless you Google it. So I would encourage you to Google it. It's pretty cool. Okay, the last type of specialized feeding is filter feeding. Most amphibians and reptiles I don't know of any adult amphibian or reptile that uses that strategy for gathering food. You might think, if you think of one, let me know, but I can't think of any. So it's pretty common in tadpoles, larval stages of most frogs. And so, just like I said, as far as I know, no reptiles or adult amphibians use this. The main difference here is that most adult amphibian reptiles feed on animals. And so filter feeding works on animals if what? Think of animals that you know that are filter feeders. Yeah, very good. Microscopic or really small animals, that works great. If you're a paddlefish, you open up your mouth and you've got really, you know, gill rakers that are close together, you're gonna filter microorganisms and then you transfer that to the gut. So it works great if you're feeding on small animals, but if you're feeding on larger animals, then filter feeding doesn't work. If you're feeding on algae and protists, then filter feeding is gonna work to some extent. So these guys mostly feed on periphyton, which is mostly algae or diatoms, and then it could be fungal organisms, bacterial organisms that grow on the surface. So peri means around the perimeter, around the edge or on the surface, and then phyton refers to plant-like organisms. And so these guys are feeding on aquatic plants, basically microscopic aquatic plants. And so, and they've got some features that allow them to do that. They have a keratin-based beak, and so keratin-based, made of keratin, same stuff that your fingernails are made of. And this is the beak, and so if you look at the mouth of a tadpole, you'll see something like that. And that's what they use to scrape the periphyton off the surface of either a rock if they're in a stream, or you know, sticks and large woody debris, or just the surface of the sediment, surface of a leaf if they're living in like a ephemeral pool in the forest. And so that's part of the mechanism for getting the algae off of the structure. And then they also have buccal papillae, which are these little extensions around the mouth, and mostly they're sensitive to chemical cues. And so as they're moving around, the epithelial cells are sensitive to certain chemical cues, and that's going to send the signal, okay, this leaf is rich in periphyton, so get going on it. And so that's what the buccal papillae are all about, and that's what allows them to identify their prey. And then once the material has been ingested, then they have structures kind of like the gill arches, or the gill arches in fishes, that help them collect the algae that get into the buccal cavity, and then that gets directed into the digestive system. And then of course they're getting nutrition from that. All the clean water comes out across the gills, which is great for respiration, and then out through the spiracle that we talked about before. And here's a group of tadpoles so you don't have to have rocks. You know, because even if you think about around here, as the water floods into, you know, grassy, kind of fieldy kind of habitat, if you don't disturb it, it's pretty clear because it's not very deep, right? So all that fine sediment settles out, and you got clear water, which allows sunlight to penetrate it, and then on the surface, you're going to get algae growing, and these animals can feed pretty heavily on that. So it's pretty effective, you know, if you have the right mechanism. Okay, does anybody have any questions on that? Okay, so your exam for next Monday ends, material ends right now. Yep.
Ends right there. It was just a question about tadpoles. You know when you flip them over,
Yeah.
you can see their belly, and they've got that circular, what's that?
That is their digestive, that's their digestive tract. Yeah, so, I mentioned that for some reason or
That was
another.
on our first exam.
It was on the first exam.
Oh, really?
Just because I was distracted with tadpoles. Okay. Yeah, so, yeah, we talked about that on the first day of class. No, not frogs. Yeah, they have a really long digestive system because they're eating mostly plant material, which is hard to digest, so it takes a long time in the digestive
Stop
system.
it, I got that question wrong, I said to fit more bugs in their tummy, because I didn't know, but that whole circle thing I've been looking at was their long digestive system, and why do they have it?
Why do they have a long digestion? Because it takes longer for plant material to be digested
Okay.
in animals, in general.
Right.
Yeah, so even like with fish species, or, you know, like,
Cows.
like cows.
Yeah.
I mean, they're gonna, you know, swallow it, grind it up, spit it up, chew it, grind it. It takes a long time to break down plant. For animals, it's hard to digest plant stuff. And so, and, you know, in human plant material diet, in your diet is called roughage, basically, because you don't digest it. It just goes right through your digestion. So if you're an animal that's relying on that, then you need a longer digestive system for it to absorb nutrients from.
It's mainly because they have to transform the plant-based energy source to an animal-based.
Gotcha.
Well, it's mostly because there's a lot of cellulose in it, and that's hard for us to digest. So even animals that eat only, like a termite that eats only wood,
Right.
they can't digest it either, but they have some symbiotic bacterial organisms that do it.
Which makes.
So do sapsuckers. You have a question?
Are Sicilians also filter feeders?
Sicilians?
Or I'm sure it might range, but
Sicilians?
yes.
No, they're terrestrial animals. Sicilians?
They're fossorial.
They're fossorial, yeah. No, they eat mostly like annelids and other small animals that might be in the set now. Okay. All right, so this stuff, what I'm getting ready to lay on you, will be on the final exam, but not on exam 3. Is everybody straight on what's going on?
There's 2 exams left.
Yes, there's exam 3, a regular season exam, and then there's a playoff exam. Okay, so, mostly what I'm getting at on this bit is reproductive biology. But of course, there's a lot of aspects of reproductive biology. So what I did was I kind of condensed it down a little bit because there's no way we're gonna finish all of it. That's kind of what we're talking, all the stuff that we're gonna talk about is related somehow to reproductive biology in some way. So, I wanted to start off with just talking a little bit about life history, and its effects on, or its relationship, I guess, to reproductive biology. So, we're talking about life history. We're essentially putting things, like characteristics of organisms in perspective with respect to this basic idea, right? So, if we're gonna see living organisms on this earth at this point, then those things have to be accomplished very efficiently. If not, then they go extinct. And so natural selection has acted on the ability to get your offspring into the next generation. And so that obviously is gonna relate to 2 things in a very general sense. One is survival, right? Because if you don't survive, you ain't what? You're not reproducing, so that's obviously gonna have an influence on the number of offspring you leave. And then your just ability to produce offspring, right? So survival and reproduction is all about that. So a lot of the things that, you know, the features of organisms that we observe are related to making that most effective under certain circumstances. And that's kind of what we're talking about with life history. So the life history characteristics of an organism are those elements that relate to how they do it, right? How do they get their offspring into the next generation? What are the strategies that they're using, not consciously thinking about, but what are the strategies that have survived, right, through the filter of natural selection? And so a lot of the things that we typically measure as biologists relate to the life history characteristic of the organisms, and some examples of that would be related to juvenile development. So where does it happen, for example, right? Is it in the aquatic environment, or is it in the terrestrial environment? What aspects of the juvenile, how quickly does it happen, right? So all those things are under the umbrella of life history characteristics of an organism. The age and size at which an animal reaches sexual maturity, because there's different ways you can do that, right? So typically, when we're talking about ectothermic organisms like amphibians and reptiles, age and size are related, right? So if you're older, what happens? You tend to be bigger because most ectothermic organisms have what we would describe as indeterminate growth, right? Which means there's no point where they stop growing. They slow down quite a bit. But for endotherms, you know, for a bird, for example, sometimes, you know, the fledgling is bigger than the adult. So they're gonna lose size as they go from fledgling to an adult, and they're not gonna get any bigger once they really reach that stage. So for a lot of organisms, a lot of the ones that we're talking about, age and size at sexual maturity age and size are related, but there's different strategies you can use to get your offspring. Right, you can reproduce younger, but then you'd be smaller. Right, what's the disadvantage of that?
Can't produce as many.
You can't produce as many because there's not enough room, right? So if you're a small female, you can only put so many eggs in your body cavity, and if you're smaller, you can put less than you can if you're bigger, right? So some organisms tend to reproduce younger, even though there's a disadvantage of that. What's the advantage of producing younger, reproducing younger?
More chances of reproduction.
More chances you're actually gonna get to do it, right? So if you wait 25 years, like in the case of a sturgeon, your chances of surviving that long are a lot less than they are if you wait 1 year, right? So, but you're smaller, so there's some advantages of growing big. So what I'm talking about with life history is that we're looking at aspects of these organisms and trying to figure out what things affect those characteristics, right? So you might see in a particular species a range of that occur. And so to understand what's the cause of that, we need to know that it occurs, it's variable, and what factors are different when we look at this population compared to that population. So sometimes within a species, you're gonna get sexual maturity at a younger age, depending on the surrounding environment, right? So if there's more predators, maybe they're gonna do that. The number and size of offspring, kind of same basic idea. On two ends of the extreme, if we look at these life history characteristics, some organisms are going to produce lots of offspring, in which case they tend to be what size? Small. Or a few offspring, in which case they tend to be much larger, right? So there's some advantages and disadvantages to those things. If you produce larger offspring, what's the advantage?
Not as well.
How come?
They're not as at risk of predation.
Right, because these animals are vulnerable to predators that generally are gonna swallow them whole, right? Gate limited is what we call that. If you're bigger, then you've already excluded a whole bunch of animals that can't fit you in the map, right? So there's advantages and disadvantages, and so life history is the study of life history is understanding what factors influence those things. Level and type of parental care is variable and falls under the category of life history. So some animals have zero parental care, some animals have extensive parental care, and it varies sometimes in a single species depending on the environmental circumstances. So those are the kinds of things that we mean when we're talking about the life history of an organism. Those life history characteristics are these things that are variable sometimes across species, but ultimately they're leading to a more efficient way of producing offspring and getting them into the next generation. Okay, so what I want to try to do is kind of link reproductive biology to those concepts, kind of life history strategies and characteristics. As soon as I figure out where I'm at here. Okay, so one of the first things that we have to think about is what happens when we talk about the transition from the aquatic ancestors to terrestrial adult, right? There's some pressure to modify what you're doing because of the drastic difference in environmental conditions. And so some of the things that we recognize that mostly transitioned, right, from the ancestral forms to the derived terrestrial ones relate to fertilization. So if you're an aquatic organism, typically, what kind of fertilization are we using, external or internal?
External.
External, how come?
There's no water there, so they
Huh?
can, they can get to each other.
Let's look at some water.
What can get to each other?
The egg and sperm.
That's right. And it's, what I was thinking was, it's easy, right? Because the ovum can be released into the aquatic environment where it's mostly water. The sperm can be released into the aquatic environment because it's water, and living organisms are mostly water. So being in water is easy for these things, and so typically, aquatic organisms tend to have external fertilization. We're talking about fish ancestry. Whereas terrestrial animals, because you can't just release sperm or an ova onto dry land because it dries out like that, because these, you don't have the protection from that. And so generally, we're talking about internal fertilization. And so most amphibians and reptiles have internal fertilization. That's not always true. Of course, there are some, like frogs, particularly most frogs, that can have external fertilization, and we'll talk about that. But that's, one of the things that typically is transitions as we talk about the aquatic ancestors in comparison to the terrestrial derived forms. Direct development is another thing that happens as we make this transition. So what that means is there's no larval stages, right? So of course, larval stages typically are aquatic, and so frogs have aquatic larval stages. Some salamanders have aquatic larval stages, but others have direct development. They don't have a larval stage, or the larval stage is completed within the egg capsule, which is more or less protected. Another one of the big transitions is the evolution of the amniotic egg, right? So we're talking about reptiles now in which the eggs have a protective, or the embryos have a protective membrane that keeps the moisture, and that just frees you up. Now we don't have to be aquatic, right? Now we can be totally terrestrial because the embryo is protected from drying out. And so, if we think about all these factors that are making the transition, as we talk about ancestral aquatic organisms to the terrestrial ones, if we look at terrestrial vertebrates, there's a huge variability in strategies for getting offspring into the next generation. And there's a mixture of these things as we go through the different groups, and you'll see basically just that as we talk about them specifically. Okay, so my original version of this had a bunch of information about how gametogenesis works and the telegenesis, but we don't have time for that, so I'm gonna brief it down just to make sure everybody's on the same page. Going back to freshman biology. All right, so, we're talking about reproduction. So, basic description of reproduction is that males produce sperm cells through the process of spermatogenesis. Females produce ova through the process of oogenesis. Some other things have to happen for these to become mature, but this is the ultimate result of that. And then the zygote is ultimately will become the embryo. Okay, so just to complete this thought here, this process, the production of sperm, is via the process of meiosis. Does anybody remember that term? The other type of cell division is what? Mitosis. They're different. Somebody tell me why they're different. What's different about them?
Meiosis produces haploid cells and mitosis produces diploid cells.
From what? What's the starting cell?
A diploid cell.
For which one?
Meiosis.
There you go.
For both
I'm
of
trying to trick
them.
you.
I was like, wait a minute, yeah, both of them.
What's that? What, did somebody say something?
I was gonna say meiosis is like gametes, while mitosis is from systematic cells.
Well, no, meiosis produces gametes. This is the gamete, right? So gametogenesis. But you're on the right track. So essentially, we're talking about diploid cells that are making more cells, right? So those are the germ cells that are producing these haploid cells. And these are going to be the gametes, right? If you're a male and you do that, that happens at the testes, then that gamete is a sperm cell. If you're a female that does that, that happens where? Come on, ladies. Ovaries, yeah, and then that's gonna be an ovum, and then they get together. So this process involves diploid cells that are making diploids. This process involves diploid cells that are making diploid cells. And that happens every time you have to make new cells, like if you injure yourself and the cells replicating. Or it also happens here, right? And does what? I'll give you one word, tell me what meiosis does to the zygote. Growth. Good. Yeah, you got one, oh, you got one word, nice. Growth, right, that's how it gets bigger. Add new cells that are exactly the same until that individual goes through sexual maturity and start producing gametes. So, okay, so what we've done here, so now what I want you to do is tell me the difference between a haploid cell and diploid cell. And it better be good.
A diploid cell has 2 sets of chromosomes, a haploid cell only has 1 set of chromosomes.
A diploid cell has 2 sets of chromosomes, a haploid cell only has 1 set, but you need to tell me how those sets are divided. Use some more terms. Use one particular term that will explain all of that.
Because your meiosis 1 and meiosis 2?
No, that's just how we describe
They're
it.
in pairs?
Well, yeah, pairs of,
Sisters.
Chromosomes.
not sister chromosomes. But you're not, well, sister chromatids are part of it. Y'all will make me do meiosis on the board.
Haploid has the dad chromosomes and the mom chromosomes.
And those 2 chromosomes are blank to each other. Homologous. They're homologous chromosomes, right? So you can't, so this diploid cell is 2N, and this is N. So if you're a human, this is 46, and this is 23. But you can divide 46 chromosomes in a bunch of different ways and get 2 sets of 23, right? You have to divide them in a particular way, and that is to separate the homologous chromosomes into different cells. Make sure you got one of each homologous chromosome, because homologous chromosomes, oh boy. What makes them homologous? They have what? Same. Same what?
DNA.
Same color?
Not DNA, but the genes.
They're
Same.
coding for the same gene.
Same genes, there you go. They've got the same genes, right? So in other words, sections of DNA that produce a particular type of protein. So you have to have some of that protein in there, otherwise whatever it does is not going to be done, and the organism is not going to survive that. So this process is gametogenesis, and then this is fertilization, and then this is the zygote, which is the embryo. And so the embryo is going to have 46 chromosomes, and then it's going to start dividing by the process of mitosis, and all the cells are going to have 46 chromosomes, and they're all going to have two copies of the same gene. So homologous chromosome, that's the main thing, right? So when this process happens, you separate homologous chromosomes into 2 separate cells, and so one of those is going to fertilize or get fertilized, and that's going to result in a diploid. Okay, so that's my quick and dirty review of meiosis. And so now we're talking about how does that happen, right? So different organisms have a whole variety of ways of making that happen. Okay, so here's just a PowerPoint version of that. I just want to make sure you think about, you understand what's happening. And some of those terms that we talked about that we shared with you guys when you were freshmen in genetics, homologous chromosomes, those are all important terms because they mean something really specific, right? A sister chromatid is another term. That means something different though, and it has an influence on what happens to the genetic composition of the organism. Okay, so fertilization essentially is that when you have a sperm cell that unites with an ovum, and each one of those is a haploid, that produces a diploid zygote. And then there's different types of fertilization, so that can take place externally in the environment, or it can take internally in the reproductive tract of the female. And so different animals have different approaches to that, and even that can be different. Actually, that's not ever true. Oviduct is the correct term for that. Okay, I'm gonna stop here, and then